fatty acyl CoA will react with the carnitine by using carnitine acyl transferase 1, and we will have fatty acid carnitine in the outer mitochondrial membrane, that is transported to the inner mitochondrial membrane by translocase enzyme. Then in the inner mitochondrial membrane by another enzyme that's called carnitine acyl transferase 2 will release fatty acyl CoA. This fatty acyl CoA actually will go to and enter the beta oxidation. Okay? So the first step is activation in the cytosol, and two high energy phosphate bonds are hydrolyzed. Okay? And this diagram is showing here we can see in the cytosol, in the cytosolic side, we do have fatty acids with carnitine. Okay, fatty acid first with ATP by fatty acyl CoA synthetase will produce fatty acyl CoA, then by translocase will enter from the outer mitochondrial membrane to the inner mitochondrial membrane, and by the second enzyme that's called carnitine acyl transferase 2 fatty acyl CoA is released in the inner mitochondrial membrane, where we can find the rest of the enzymes of beta oxidation. Okay, so the enzyme acyl carnitine transferase 1, 2, and translocase and fatty acyl synthetase enzymes. Then beta oxidation are series of reactions. It's actually a series of 4 reactions. The first reaction is dehydrogenation or oxidation. The second reaction is hydration, then oxidation, dehydrogenation, then thiolysis. These series of reactions, generally, you don't need to know details about them. The only enzyme that you need to know about is fatty acyl CoA dehydrogenase. It's one of the very important enzymes in these 4 steps that you need to know. When the fatty acid enter these 4 steps, will produce, acetyl CoA, which is 2 carbon units, and FADH2 and NADH. So we'll need FAD and we'll need NAD, where this reaction will produce FADH2, NADH, and it will release 1-acetyl CoA. So if we started the oxidation by 16 carbon units, in the first turn we will have 1-acetyl CoA, which is 2 carbon unit, and we will have 5 ATP, which come from the FADH2 will produce 2 ATP, as you know, and NADH will produce 3 ATP. So in the single turn, we'll have acetyl CoA will have 5 ATPs, from FADH2 2 ATP and from NADH 3 ATP. So the reactions are dehydrogenation, hydration, oxidation, and thiolysis. When we look at this diagram, we can see that from C16 to C14, we will have 1-acetyl CoA. From C14 to C12, we will have acetyl-CoA. Another acetyl-CoA can be produced from C12 to C10 and C10 to C8, C8 to C6, C6 to C4, and then two acetyl-CoA can be released or can be formed. So when we have odd chain fatty acids, this means that from oxidation of the odd chain fatty acids, we will have propionyl CoA, which is then can be converted to another biomolecule that's called succinyl CoA. And as you know, the succinyl CoA is one of the intermediates of the tricarboxylic acid cycle. Okay, the alpha oxidation is also seen in the brain. Removal of one carbon at a time from the carboxyl terminal. Generally, this is the idea of the alpha oxidation. Omega oxidation is a very minor pathway and it requires cytochrome P450 for the hydroxylation, and generally it's occurring in the endoplasmic reticulum. Beta oxidation generally has some clinical applications. First, medium chain and short chain fatty acids do not require, as we mentioned before, carnitine for transport across the mitochondrial membrane. So, medium chain and short chain fatty acids are easily oxidized. But sometimes we do have carnitine deficiency, and this is reported, and thus we will have more glucose is utilized, and this will lead to hypoglycemia. Also, translocase can be deficient, and sometimes the carnitine palmitoyl transferase 1 can be deficient also and it will lead to one of the clinical applications. Thank you.